Before you start, could you also please introduce yourself with a couple of sentences?
Okay, I can of course do that. So basically, I'm a group leader at MPI CBG in Dresden, and the topic of my lab you could basically describe as membrane chemical biology. So we design probes to try and understand lipid function, and one of the key aspects in the lab is to understand how lipids function in signaling. So for my background, I'm a chemist by training and studied in Göttingen. And then I basically moved into cell biology at EMBL in Heidelberg, where I worked as a postdoc in the lab of Carsten Schulz, and then I became PI here in Dresden at the Institute.
Thank you very much, André. So you can start sharing your screen.
I did.
We are looking forward. Okay,
so you're seeing my screen, no?
No, I can't.
Okay, because I did share it already, I think. Okay. Next attempt.
Something else. Yep.
Okay. Good. Okay, so today's seminar will be about a paper that we recently published in PNAS, and I want to point out that the first author, Milena Schumacher, is an extremely fantastic student, very creative, and is currently on the job market, so she's looking for her next position. So as a way of introduction, I'm going with a little bit of a provocative statement. The way we think about lipids in cell biology is usually very simplistic. So we draw up these cartoons where we have little heads and then some wiggly tails, and then we make up a membrane. And even this schematic structure here implies something about lipids that is not necessarily true, that we essentially have a head group and two fatty acid tails. But if we look at what kind of solutions nature has found, sorry, that's my telephone. Somebody apparently is extremely important. Nature has found to make up, to create membranes. We have, of course, these classical. Kann ich bitte zurückrufen? Ich gebe gerade einen Talk. Okay, fantastic. Thanks. Thanks. Bye-bye. Okay, that was one of our directors with an appointment for tomorrow. So it's very clear.
I hope you still have the job.
Yeah, I mean, I have, I think, by now. Okay, so we have classical glycerophospholipids and sphingolipids that are the major species, structural lipids and signaling lipids in eukaryotes. We have sterols that are also primarily a eukaryotic lipid. Then the bacterial analog in the form of hoponoids. And if James Science is listening in, shout out to him. We have rather complicated glycolipid structures like lipid A. And then there's these weird molecules in archaea where you essentially have head groups at both ends. Also, you don't have a traditional bilayer anymore. Also, this is essentially an amazingly complex heterogeneity in terms of chemical structure that you see. And one thing that it makes extremely clear is that there are multiple solutions to make a functional membrane. Also, the structure in the lipidome is not as conserved and not as regular as you would find it in the proteome or even for nucleic acids. But this is not everything. There's something that basically gets more and more appreciated in the last 10 to 15 years due to a multitude of lipidomic screens that were carried out. And that is that even if you look at just one lipid class, in this case, phosphatidylcholine, which is the major structural lipid in mammalian membranes, what you see is that this is not a single species as depicted here, and as you would do it typically in a review article, but you have at least 30 to 40 chemically distinct species that are present in each organism. And one of the big mysteries of our time in lipid research is what are actually the functional implications of this so-called lipid diversity. Now, how does it come about? The first thing is that fatty acids can be attached differently to the glycerol backbone. You can have ester linkages here. You can also have ether or vinyl ether combinations. You can have hydroxylations where, for instance, double bonds are hydroxylated. The double bonds themselves can be different in number and in positioning. The chain length can differ, and you can have aldehyde groups if there's an oxidative cleavage of the double bond. And essentially, the key question is, why does the cell do that? What is the function of this amazing diversity that we find in the cellular lipidome? And I think a good way to think about this is to take a step back. And here I'm borrowing a concept from Gerrit van Meer and try to break down lipid function into major categories. Is lipid diversity required for membrane function, membrane formation? And I would argue not necessarily, because you can make a perfectly fine membrane with just one lipid species. And this, I don't think you need thousands of individual lipid species for that. Then there's energy storage, where you have triacylglycerides that essentially are stored away in lipid droplets. And then there's this large area of signaling where lipid play important roles as first and second messenger. And I think this is where we can argue that the cell would benefit a lot from having multiple species there that would allow it to fine-tune its signaling pathways. Now, the example that we're going to look at today is diacylglycerol signaling, which is arguably the most prominent class of lipid second messengers. And if you look at how they are typically generated in the cell, is that you have in the plasma membrane a relatively high content of about 0.5% to 1% of a phosphoinositide molecule. That's depicted here, PIP2. And by action of phospholipase C enzymes that are activated upon binding of ligands to cell surface receptors, the second messenger diacylglycerol is generated. So essentially what happens is that this phosphodiester bond here is cleaved. You generate the glycerolipid diacylglycerol, IP3, which is essentially just the PIP2 head group. This then induces calcium signaling, but diacylglycerol itself has also important roles in signaling. Now, you take any review in the field, and you get, when you look at the signaling network, and what you get is something like this, where you essentially have inputs from the cell surface, from growth factors, from GPCR ligands that activate their respective receptors. This triggers PIP2 cleavage, diacylglycerol is generated, and then you get this important arrow to signaling effectors. We're going to talk about a little bit more about what these proteins are. And then you get an entire array, a host of responses by the cell. And some of them are directly contradictory. So you have apoptosis and proliferation. And not a lot has changed in these figures. So basically, it has been shown that diacylglycerol is in one way or the other implicated in all these processes. And the point about these kind of networks is that they essentially represent hourglass models. So you have a large variety of inputs. You have a similar large variety of outputs, but everything has to be shuttled through this bottleneck of the second messenger. So information that comes from the outside has to be processed through this entire signaling cascade, and also has to pass through a single entity here. Now, you can look at the math behind these systems, and if you do an information theoretical analysis, what you will find, and what people have actually shown and proven, is that the information processing capacity of such networks is exceedingly low once you take into account biological noise. Once you really think about what it means that every cell is different, that you have different expression levels of effector proteins, that not every cell can generate the same amount of second messenger and so on. And in a number of studies, people have repeatedly pointed out that the bit depth, like the number of different messages that you can send through such a network, is essentially 1. No? So we know this cannot be true. So there needs to be something that allows has to allow these networks to have better information processing capacity. Well, simply because we know phenomenologically that signaling works. So the cell knows what it does, but it also means that our understanding of the signaling network is limited. Now, one idea that we've tried to pursue in the last couple of years is that actually the cell uses the structural diversity in lipid second messengers to encode information. And this would essentially create a situation where you don't have an hourglass model anymore, but something that is more representative of a filter bank, where you don't have a single lipid second messenger species, but more of an array of chemically different species that then could be used in combinations and linear combinations to encode different messages. How do you begin to test such a hypothesis? One thing that is exceedingly clear is that basically everything that I've drawn up here as these lines are biochemical reactions. Also, to understand how different the reaction rates are, the affinities are that can be encoded in a lipid second messenger spectrum, what we need to do is to do quantitative reaction kinetics in the living cell. And this is essentially what I'm going to talk to you about today. What is a kinetic relaxation experiment? So typically, if you go in vitro and try to understand how a reaction mechanism works, a kinetic experiment, very schematically drawn up here, is where you have an initial equilibrium or a steady state of the system under investigation. And what you do is that you basically introduce a perturbation on a time scale that is much, much faster than any process that you want to analyze. And what this results in is basically that the system wants to adapt a new equilibrium, a new steady state that's drawn up here, and it needs a certain time to reach this new state. And this is usually in an exponential relaxation process. And essentially, this process here gives you the information about the reaction kinetics. There's a number of ways this is usually implemented in physical chemistry. The most straightforward experiments are, for instance, pressure or temperature jump experiments, where you basically move the physical properties of the system in a very fast fashion. But this is not something that we can easily implement in live cell experiments. Another opportunity, for instance, is to use photolysis, where you basically alter the concentration of one of the reaction participants in a rapid manner. And you make use of the fact that the kinetics of photolysis reactions are usually much, much faster than any other reaction. And this is what we are going to use here. But there's a number of additional key requirements that we need. We need rapid perturbations, and they need to be local and on the level of individual lipid species. What do I mean with local? In this case, I mean that it has to be in one membrane of the cell and can't be in the entire cell. And the other thing is that we need fluorescence microscopy as a readout to get molecular information. Now, how do we begin to, what are the tools that we're using for that? And one of our most successful tool sets is the use of so-called photocage lipids, which can be used to locally change the level of lipids in the cell. And you can imagine this as that we take in the chemistry lab a native lipid species, and we attach something that we call a caging group. So, for instance, you can use one of these coumarins here and attach it to a position on the head group of the lipid that it's crucial for its biological function. And what this now results in is that, for instance, its interaction with proteins is blocked, and you essentially have an inert molecule sitting in the membrane of the cell. Once you have it in the cell, what you can do is you apply a flash of light. This removes this caging group and gives you back these native lipid species. And the result of this process is essentially a stepwise, like in a millisecond range, a stepwise increase of the lipid concentration in your membrane of choice. There's another thing. These coumarin groups here are fluorescent, and that means that you can easily monitor where your precursor molecules are. We use yet another trick before we go to actual data. We put these sulfonate groups on, and this allows us basically to load the caged lipid probes only in the outer leaflet of the plasma membrane. And the driving force for that is that the fatty acid side chains get incorporated into the hydrophobic part of the outer leaflet, but the sulfonates on the coumarin group, due to their intrinsic negative charge, completely block lipid flip-flop. So prior to uncaging, these molecules reside in the outer leaflet only. Oh, and only after cleavage of the coumarin group, they can begin to be moved to the inner leaflet. So the compound set that we use is 3 different diacylglycerols. So we use 1 short chain diacylglycerol as one extreme point. So this has 2 C8 chains. Then we use the so-called SAG, stearyl arachidonyl glycerol. And this has 4 double bonds in a C20 chain and a C18 zero chain. And stearyl oleoyl glycerol, which just has a single double bond and a saturated second chain. So this essentially gives us 3 distinct combinations: a short chain saturated one, a long chain very unsaturated one, and a long chain almost unsaturated, almost saturated species. This molecule on the right here is the regioisomer, and this is our negative control. So here the caging group is attached not at one of the primary OH groups where you have the fatty acids in this case, but on the secondary OH group. And now this head group, the symmetrical head group that you see here, is not able to bind to like 1,2 diacylglycerol effector proteins. So this is a biologically inactive molecule, but it allows us to replicate the photoreaction and thus serve as a proper negative control. Now, the first order of business was to find out whether these molecules actually localize to the correct intracellular location. And so this is basically a fluorescence microscopy image of caged stearyl arachidonyl glycerol, and you can see that it nicely localizes to the plasma membrane. The next thing is to do proper kinetics, we actually need quantitative data. So we need to know in absolute terms how much of this compound ends up in the plasma membrane. Well, so we really need to measure how many molecules we do have per square micron. And this we did by first generating plasma membrane maps from cells that were loaded with the caged diacylglycerols. And we did this to basically have a defined 3D structure of that we can actually know that we're only looking at a single membrane and not as with the cell, we have no idea how the membrane density actually is. And then next we prepared GOVs, artificial vesicles with defined content in the lipid mixture of the caged lipid probe. So this was not only input, but we also checked by mass spec that we ended up with the correct concentration. And what you then can do is that you can draw up a calibration curve and determine how much caged compound you have in the plasma membrane of living cells. So this is something about 0.04% of total lipids. And what this means is that the probe content in the plasma membrane is roughly 1 in 2,000 lipid molecules. This is something that I want to highlight because we often get the question whether this actually has structural implications for the membrane. And I think if you have a content that is so low, this is something that you can rule out. It's also something to keep in mind about the limitations of this approach. This is very good when you discuss processes that only require tiny changes of the concentration of one particular species, as is the case for signaling. But I think it's more limited when you really want to modulate structural properties in the membrane, where you would have to change lipid levels in the single percentage or two-digit percentage range. Now, the next thing is this only gives us the number of probe molecules incorporated into the plasma membrane. But what we're really interested in is how many actual active diacylglycerol molecules are liberated. And for this, we use what I would call a dirty chemist's trick. So if you wait long enough, a couple of hours, what happens is that part of the plasma membrane gets endocytosed. And that, of course, means that also a little bit of these caged lipid probes ends up in endosomes. But importantly, the coumarin fluorophore will be on the inner leaflet of these endosomal structures. So now if you do the photoreaction, what happens is that from the molecules on the plasma membrane, the coumarin fluorophore gets released into the extracellular space. So that disappears simply because it diffuses away. But in the endosomes, the coumarin gets released into the lumen of the vesicles, and it stays fluorescent. So you can use the drop of the fluorescence at the plasma membrane to calculate the relative amount of photoreleased lipid, and you can use the fluorescence in the endosomes as a control for unspecific bleaching. And on the left, you basically see an experiment, how this looks like. After photoactivation, which essentially happens in the middle of this looping movie, plasma membrane goes dark, but the vesicles stay fluorescent. You can quantify that by a segmentation tool, and then essentially this down here is the drop in mean fluorescence intensity, and this gives you the relative photocleavage efficiency. And now, since we know the number of caged diacylglycerols in the membranes and the density, we can exactly calculate how many of these molecules get photoreleased. The next part is to try and figure out what kind of effects we see on actual signaling. And for this, we turn to diacylglycerol effector proteins. And the most prominent diacylglycerol effector proteins are so-called protein kinase C isoforms. Diacylglycerol reactive protein kinase C isoforms come in two flavors. You have novel PKCs, which essentially have tandem C1 domains, and these are the domains that actually bind diacylglycerol and recruit the PKC to the plasma membrane. And then you have classical PKC domains, which have an additional membrane binding motif in together with the C1 domains. That's the so-called C2 domain, which binds calcium and anionic phospholipids. So these proteins, these kinases, have a much more complicated membrane recruitment mechanism. So we monitored PKC activation by monitoring its intracellular fluorescence. And before uncaging, these proteins are largely cytosolic, as you can see here on the right, where cells transfected with PKC epsilon EGFP are pictured. And then after uncaging, where the diacylglycerol content on in the plasma membrane increases, these proteins get recruited to the plasma membrane, and then you can calculate something that we call the translocation efficiency. This is essentially a ratio of normalized fluorescence intensity at the membrane and in the cytosol. Now, a typical experiment would look then as follows. So you have a baseline, diacylglycerol gets uncaged, and then the protein gets recruited to the plasma membrane, but simultaneously metabolism and lipid transport kicks in, which then reduces the content of diacylglycerol in the plasma membrane again. So you get a pretty stereotypic phenotype where you get an increase at the plasma membrane and then a drop again. The next thing is that diacylglycerol is not only recruiting protein kinase C isoforms to the plasma membrane, it also activates plasma membrane calcium channels. And typically these are TRPC 3 and 6. So it was the obvious thing to do to simultaneously measure protein kinase C recruitment and calcium transients. And the data that I'm going to show you in the following, and also the quantifications, are typically two-color experiments where we monitor in the same experiment protein kinase C recruitment and calcium. And this is now an example where we are looking at PKC alpha, so that's a classical isoform. And in the red channel, we have an intensometric calcium sensor. So an increase in fluorescence corresponds to increased intracellular calcium levels. This is how it looks like. You get recruitment of the PKC alpha to the plasma membrane, and simultaneously you get a calcium burst that then recedes again. So how do the quantifications look like for the individual PKC species? So this is going to be a very complicated figure, but it has a lot of information in it. So what we did here is that for each PKC isoform, we did a large number of diacylglycerol uncaging experiments. And the colors that you see here stand for the different diacylglycerol species. So pink is the highly unsaturated diacylglycerol species. Green is the long chain diacylglycerol species that only has one double bond. And blue is the short chain variant. Gray is always the negative control lipid. And for PKC epsilon, you can see that there's a very clear graded response. So the highest amplitudes are found for SAG. Then you have an intermediate response for SOG, and the short chain variant has the smallest response. Whereas for the negative control lipid, we don't see anything at all. And then basically you can play this game for each of the PKC isoforms. What you see is that each of these molecules has a different response pattern to the different diacylglycerols. And one particularly interesting aspect is, for instance, if you take the pair of PKC alpha and PKC beta, which are both very closely related classical isoforms, is that you have opposing reactions to SAG and SOG. So basically the pattern that we observe here completely flips. So there seems to be a lot of selectivity in this patterns that we find. What I want to stress here, especially for this PKC alpha, PKC beta pair, is that the calcium signals that underlie the recruitment of the protein kinase C are largely superimposable. So what we think is that this effect that we see here is really something that is transpiring because of the chemical differences in the diacylglycerols. So basically, the major take-home messages. So I don't intend to you for remembering all these details. The major take-home messages are that you get different recruitment patterns for all of the diacylglycerols that we tested and individual PKC isoforms. So this is a pretty strong indication that our initial hypothesis is actually correct. And then the second thing is that the short chain analog, deoctanyl glycerol, this is a commonly used tool in cell biology. In our hands, when you activate the exact same amount as for the long chain lipids, this doesn't recruit classical PKCs at all, and it doesn't induce calcium transients. Also, basically, this analog doesn't recapitulate what native diacylglycerol species do. The next thing that we were wondering about is how closely does this actually resemble the patterns that we would see after activation of physiological diacylglycerol production. Now, you keep in mind that physiological diacylglycerol is generated by action of phospholipase C on PIP2. And the typical resting state fatty acid composition of PIP2 is heavily skewed to species that are highly unsaturated, like the stearyl arachidonyl species that I've been talking about so much in the last minutes. And this is actually the lipid fatty acid composition that is archetypical for phosphoinositides. So what we would expect to find is that physiological diacylglycerol production should resemble what we find for this highly unsaturated diacylglycerols, and not so much for the more saturated species. Now, I've replotted here the pattern that we find for the different diacylglycerol species, and in pink you find highlighted the pattern for SAG. So you get a strong and pronounced response recruitment of PKC epsilon, a briefer recruitment with an intermediate amplitude for PKC delta, almost nothing for PKC gamma, strong response for PKC beta, and only a small response for PKC alpha. Now, if we take the same cells and activate purinergic receptors by simple addition of ATP, we find something that is essentially a mirror image. So you get like only little response in recruitment of PKC alpha. PKC beta recruitment is stronger, only little for PKC gamma, intermediate response for PKC delta, and a strong response for PKC epsilon. So this essentially gives us a lot of trust that what we are doing is actually reflective of what happens in the cell. Now, I've promised you mechanisms, because so far this is a quantitative experiment, but a relative comparison as a readout. If you draw up the simplest kinetic scheme that you can come up with to describe a lipid signaling event with diacylglycerol as the second messenger, what you have is something like shown here on the left. So you have different lipid pools. You have a pool of diacylglycerol on the outer leaflet of the plasma membrane. You have a pool of diacylglycerol of the inner leaflet of the plasma membrane. You have a pool that is bound to the effector protein. And of course, you have a cytoplasmic pool of the effector protein that is not lipid bound. There's a number of rate constants that basically describe the interconversion of these different pools into each other. You have a rate constant that essentially describes the flop reaction, so from the inner leaflet to the outer leaflet. You have one that describes the flip reaction from the outer leaflet to the inner leaflet. You have an affinity between the lipid and the protein. And then you have a rate constant that essentially summarizes lipid metabolism, lipid transport. In essence, every process that removes this lipid from the inner leaflet of the plasma membrane. Now, if we would do this with a full-length effector protein, we would also have to take into account cooperativity, so lipid binding to individual lipid binding domains within the protein, and also interaction with further proteins down the signaling pathway. So what we decided to do to further simplify the system and go with a minimal diacylglycerol binding protein. So this is essentially just a single C1 domain, fluorescent tag, EGFP, and a nuclear export signal that basically eliminates nuclear pools. Because if this wouldn't be here, what we would be doing is to create a very expensive way to measure nuclear export kinetics, which is essentially what we did first prior to improving our lipid biosensors. Now, a typical experiment looks as follows. You basically have this protein in the cytoplasm. You do your uncaging experiment. The C1 domain is recruited to the plasma membrane and then slowly dissipates again as metabolism and lipid transfer kick in. Now you do that for the individual diacylglycerol species, and what we find is that the amplitudes become much more similar as compared to the full-length effector protein. So part of the selectivity apparently is encoded in these arrays of multiple lipid binding domains. However, the kinetics of these curves look similar, look significantly different, so that we would be able to conclude upon the rate constants that essentially are only lipid dependent, for instance, the flipping rates. To do this quantitatively, what we did is to recombinantly express the C1 EGFP nest construct. We then acquired essentially again a calibration curve And using this and always using the same settings for the uncaging experiments, what you can then do is to determine the concentration of this lipid binding protein in each individual cell. We chose to monitor the concentration of the free protein in the time trace data, as this is what is most easiest accessible by image analysis. And a typical time trace looks as follows. So on the left, basically what is plotted here is the protein molecules per cell. So this is an absolute number. You have your baseline, and this is the cytosolic number. You have your baseline, and then the number of cytosolic proteins drops until it reaches a minimum and then slowly recovers. And this is indicative of a mirror image increase at the plasma membrane, which you can then simply calculate by mass conservation. And to these traces, because these are absolute numbers, you can then fit a quantitative kinetic model. And in this graph here, you basically see the fit to the data. And I'm playing a little trick because in the model, if you remember, I had at least 5 free parameters. So what we did was not only to have one time trace to fit these data to, we varied the laser power, generated an entire measured disoglycerol liberation for each of them, and then acquired time trace data for each laser intensity, where we liberated a defined number of disoglycerol molecules, and then did a global fit to all of these molecules at the same time, to all of these conditions at the same time. Well, and then basically out of these populations, you get actual true affinities and rate constants for the different disoglycerols within the context of this particular model. And what we find here is that the flipping rates largely differ by a lot. So the long chain lipids have a much slower in rate as compared to the short chain lipid, and this kind of intuitively makes sense, no? Because this is a much smaller molecule and probably can pass across the membrane much faster. The thing that came as a surprise to us and something that we didn't quite understand initially is that the highly unsaturated species, steroid arachidoglycerol, has a much higher outrate as compared to the more saturated species, steroid oligoglycerol. However, a couple of months ago, a paper from Ilya Leventhal's lab came out where they looked at the asymmetry of the plasma membrane. And what they find is that highly unsaturated lipids, such as the one here, are more concentrated on the inner leaflet of the plasma membrane. So what you have there is an intrinsic thermodynamic driving force for highly unsaturated lipids to move to the outer leaflet of the plasma membrane, because that's a potential chemical potential gradient. No? And this is exactly what we find here. No? So you would then expect that the outrate for highly unsaturated lipids that can flip spontaneously, so where the thermal energy is high enough, is higher than for more saturated lipids. The next thing is we find that both long chain lipids are metabolized much faster than the short chain variant. And this probably makes a lot of intuitive sense again, simply because these are the native substrates for disoglycerol lipase and disoglycerol kinases, whereas this more soluble substrate is probably not. And this again is a very cautionary tale about using soluble lipid analogs in cell biological experiments, simply because the rate constants that we find here for this short chain lipid and the long chain physiological lipids differ by orders of magnitude. And lastly, the most important graph is that the KD values that we find for the lipid-protein interaction can differ by up to an order of magnitude. And this is essentially what we believe is where the selectivity in lipid signaling can come from. So to conclude, I've shown you today a new methodology. So this is an example where you basically where we've basically shown that you can, in a quantitative fashion, in an organelle-specific fashion, modulate lipid levels of distinct lipid species in single cells in lifestyle experiments. And second is an example of the possibility to determine lipid-protein affinities, lipid transbilayer dynamics of native lipid species, and lipid turnover in living cells. I have to stress here, what we do is we fit a kinetic model. We think that it makes sense to these data, but of course, you can also use different, more complicated models. It's our philosophy in the lab to always apply Occam's razor. So this is the simplest model that you can write up. And it does explain the data, so we would have to have a good reason to go with more complicated models. The next thing is that in terms of biology, that the structural variations that you have in lipid species really strongly influence the kinetic parameters, so the lipid dynamics and protein affinities. And this is the prerequisite for lipid diversity playing a role in signal specification. There's, of course, acknowledgments. The people, so the major person on this story was Milena Schumacher, shown here. Also Nico and Pavel and Annette contributed. Alexander Walter was our primary collaborator, so he's a theoretical neurologist in Berlin. We couldn't do this without the facilities at MPI CVG. And then there's, of course, the people who gave the money. I thank you for your attention, and I welcome any questions that there may be.
Thank you very much, André. Fantastic. It's an amazing tool.
Thanks.
And great presentation. So I think I will start with my questions. As
okay.
always. Since I have the power, why not?
Yeah, sure.
Okay, so first question is probably general, and probably many people are thinking already.
Mm-hmm.
So now you showed an example with diacylglycerol. Is there a limitation that we can, which lipids we can use for this kind of caging? Can we use sphingolipids, ceramides, cholesterol?
So in principle, no. So so Christina here is currently writing up a paper where we will introduce phosphatidic acid, PE, PS, cholesterol, and I'm sure I'm forgetting something. But also for single chain lipids such as LPA, this also works. Yeah, so we are putting together the manuscript, but in principle, it should be independent of what kind of lipid. There is, of course, the issue that I alluded to in the talk. This is a super nice tool set when it comes to lipids that are minor components, where you get a physiological, or where you get an effect when you change a minor fraction of the membrane. Of course, if you need to change something like 10% of total lipids, and you have you generate high energy species, which are the immediate product of this photoreaction, the likeliest outcome is that the membrane simply blows apart. No? So there is a concentration limit on what you can do. Mm-hmm.
Thank you. Okay, I'll start with the questions by the audience.
Mm-hmm.
Abhishek Kalarikar asking, can the fluidity of the cell membrane govern the quantitative changes in the signaling lipids?
Yes. So, of course, I would predict that flipping rates of diacylglycerol should be simply a function of the membrane properties, of which fluidity is one. So if you have cells with a more fluid membrane or a more stiff membrane, I would predict that the recruitment patterns of diacylglycerol effector proteins vary. And actually, we did one experiment that didn't make the final story, but was in the bioRxiv preprint, where we extracted cholesterol, and this actually changed. So in living cells, from the plasma membrane by cyclodextrin treatment, And this actually changed the recruitment patterns. So I think this is actually one mechanism how lipid signaling events can be regulated.
Okay, thank you. Okay, Ed Lyman says, really cool. And Marcus Tesano is asking, great talk. I'm a bit confused about the difference in KD values. How does the kinase sense the difference in lipid tails?
Yes, that's a very good question. Short answer is mechanistically, we don't know. We can only speculate. Well, so this is, of course, an apparent KD value that comes out of this particular model. One thing that you could think about is that some of these lipids get recruited to nanodomains in the membrane, and then you would have cooperativity, and this would, of course, alter the apparent KD value. Another one is that it could be completely straightforward, that basically the head group of the presentation of the diacylglycerols is different for each species. No? So basically, some of them might protrude a little bit further out of the membrane, some might be a little bit more hidden, and this would, of course, directly affect the KD value. But I can't tell you yet how this actually comes to place. I mean, the only thing is, these are the values that come out of the model. And even if we fiddle with what reactions we put into the model, what we always see is the same trends in apparent KD values.
Great, thank you. So next question, I'll ask one more question.
Okay.
So I already asked if there's limitation on the lipid species, but is there a limitation on, probably microscopists would ask, is there a limitation on the fluorophore that you're actually putting on the lipid? Did you test different fluorophores? And
for these coumarines, we haven't done that yet, that we put fluorophore. I mean, the thing is, its major function is to act as a caging group. So the photochemistry, so that you can basically tracelessly cleave and regenerate the native lipid, that's the major point on this. There is one paper by Dima Yushchenko and co-workers, and what they did is that they basically modified the caging group with a far-red fluorophore. Well, so essentially, you have the photocaging group, then you have a linker, and then you have a fluorophore, and this allows you to basically image these probes in one channel and do the photochemistry in the other. Well, and that's a neat little trick. Drawback is, of course, that you have one less channel to actually monitor cellular responses. So in principle, yes, you can do that.
Brilliant. Okay, so next question is by Falk Schneider from Oxford. Really, really great talk. Thank you. Can you ungauge only a little proportion of DAGs at the membrane and check how many molecules are necessary to create a response, for instance, calcium flux in the cell?
Yes, this is essentially what we did. No? So these individual traces here are for different laser powers. No? And for each laser power, so for the higher laser powers, we uncage a lot. And for the lower ones, there's almost nothing. So you can essentially really titrate the responses. No? So, and also this is a necessary requirement to actually fit the data. Because otherwise you don't get enough constraints on the model. And I think the lowest number of diacylglycerols that where we still saw a response is something like 10 to the power of -5 mol%. So it's a really tiny fraction. And it speaks to the high affinity of these diacylglycerol effector proteins.
Brilliant. So in one graph that you showed, there was also, when you were measuring K in, K out. For the K out, the saturated one was higher, but also the very unsaturated one was also higher, right? So that was exactly. So
this is the short chain lipid.
The short chain, okay. But it's still saturated, correct?
Yes. Okay, I'm going to talk about unpublished data. So we made a dimyostirene diacylglycerol.
You can tell me unpublished. I'm not going to tell anyone.
Yeah, no, it's just on YouTube. So that's the shortest chain physiological lipid. And that has much slower dynamic than this dioctanoyl. So what I think here, what we're seeing here is much more that this molecule is happy both in aqueous compartments and in the membrane. It's not a true lipid. So, and I mean, these are even lower bound estimates. Also, this thing equilibrates faster than our acquisition speed is. So what I would, so from the short chain analog, the only thing that I would take home is that this doesn't behave like a native lipid. So when you do experiments with it, you need to be aware that partitioning, dynamics, and affinities are really not what you would see for the real thing.
Mm-hmm. Thank you. Okay, next question is by Magdalena Marek. Great talk, thanks. Can you use cage lipids on organisms with cell wall?
I don't know yet. So we have a few collaborators who try that with yeast, but we haven't heard back from them yet. If she wants to do it, I'm happy to send a package.
Okay, I'm sure she will get in contact.
I don't know. I mean, it's a small molecule after all. No? So one would have to try. Mm-hmm.
Yep. Okay, next question is by Ed Lyman. He says, isn't the difference between SOG and DOG just a single double bond in one chain? Why then such a different transpilar flip-flop rates?
SOG and DOG, no. So the O stands for oleoyl in one case and octanoyl in the other. So you have a C18 chain for the oleoyl SOG and two C8 chains for the DOG. I admit that our nomenclature here can be improved. We'll come up with a better acronym in the next paper. Sorry for that. That's essentially my bad.
Okay, I think we're at the end of this questions. It's wonderful talk, André. Very, very useful too. I really enjoyed your seminar.
Thanks.
And probably I'll bother you with some other lipids.
Of course.
Thank you very much for your time.
Okay, thanks. Bye-bye.
Bye-bye. Have a great